Hello, welcome to the Society for Cryobiology webinar hosted by the International Cryobiology Young Researchers Organization, also known as ICYR. I want to thank everyone for attending. We have a really special webinar today highlighting some young cryobiologists. Um, Quick background, so ICYR is a virtual organization that we're trying to bring together young cryobiologists from across the globe and provide them with resources and events that strengthen their careers in cryobiology. Um, and we can't thank the Society for Cryobiology enough for giving us this opportunity to host this webinar. Nicole gave me some things that I need to bring up. So Cryo 2026 is going to be this summer from July 15th to the 17th in Seattle, Washington. We're going to have abstracts close for the conference on March 2nd. We have student travel grants and awards closing on February 19th. And then we have the Day Young Gao Young Investigator Awards starting March or due. Those submissions are due on March 15th. So let's go ahead and get rolling. I'm excited to introduce AKALABYA BISSOYI from Manchester University, and he's going to present on some of their novel polyampholyte cryoprotectants. So over to you, AKALABYA.
Hi. Can you guys see my presentation?
Yes, sir.
Yes. So today I'd like to present my our work on polyampholyte cryoprotectant for preservation of cells and RBC. I'm currently working with Professor Matthew Gibson in MIB, University of Manchester. So my journey as a researcher, I am from India. It's a small part of India called eastern part of India, Odisha. It's famous for it's a very good cultural place. And then where I did my PhD. After that, I went to Israel to work with Professor Ido Broslavsky for almost 3 years, and then I moved to UK to work with Professor Matt. The overview of my presentation. Today, I wanted to highlight some of our research on cryopreservation of liver cell spheroid and cryopreservation of avian blood RBCs. Then discuss about something related to how we can see ice damage to the cell using cryomicroscopy and Raman spectroscopy. So as we know, the cryopreservation is a complex process. It involves different steps like cooling the cells, adding different cryoprotectant, and then recovering the cell by thawing the cells. It involves different steps. Which also we need different cryoprotectant like intracellular cryoprotectant. We generally use DMSO or glycerol for that. It prevents the intracellular and extracellular ice formation, but there are different other things which also involve in the cryopreservation process, like ice recrystallization and also post-thaw apoptosis, which can be caused by different factors like osmotic stress, CPA concentration, and change in the lipid bilayer, membrane bilayer, and also damage in the or maybe protein denaturation. So For to solve this problem, there are different type of crop protectants are available, but a special group of crop protectant which involves which is called as polyampholyte can we studied this type of crop protectant. This is all started from one researcher from Japan. His name is Professor Matsumura. He initially proved a concept of polyampholyte, which contain both positive and negative charge, and they develop a polyampholyte which made up of carboxypolyal lysine, and it shows very effective in cryopreservation of different type of cells. The way it works, it decreases the gel liquid transition temperature of the lipid, and also inhibit the membrane leakage during the post thaw. So, based on that, in our lab, Professor Matt has developed a crab protectant or polyamphylite using bioadhesive and food additive, which we call as a magic material. And we saw that using this crab protectant, the polyamphylite, in combination with DMSO, we have better cell recovery in case of in the cells which are cryopreserved cryovials, or also in case of cells which are added into the tissue culture plate and directly cryopreserved. Then there is another problem which is long-standing problem in the field of biology or in the field of medicinal chemistry to use animal as a testing model. It is good. It is very good to predict the capability of drug molecule or testing drug toxicity or any kind of disease model, but there is ethical issue involved, and it is expensive. Similarly, two-dimensional culture, the traditional way of doing cell culture, is cheaper, it is faster, but it will not give you the accurate or the condition or the disease model that you are looking for. So spheroid is present in between them. It is not much ethical, and it is cheaper compared to two-dimensional culture, and it will give a clear indication of what you are looking for. Also, there are different advantages of spheroid. It can easily mimic the 3D tissue environment, and also it also form a gradient formation which replicate the drug and nutrient distribution of the tissue. So taking this as a note, we try to cryopreserve the spheroid on organoid using our method. What we did, we first prepared different batches of spheroid using normal standard method. It's called agar molding method, and then we try to cryopreserve them in both way. One directly using magic plate or cold sink. Put them in cold sink with your cryoprotectant solution, and directly put them in -80-degree freezer for 24 hours. And another way that we use, take the spheroid, put in the cryovials, and then cryopreserve using Mr. Frosty. In both the way we did cryopreservation, we saw that it is better than the cell viability, cell recovery is better in case of when we use different concentration of polyamphylite, especially 40 mg of polyamphylite, which significantly give us good result compared to other batches. And we did some confocal imaging to see how the cells look like in the after cryopreservation. It seems that using 10-20% of polyamphylite or 40% of polyamphylite, the cell viability is more, and the cells which are in the inner core also been cryopreserved properly. Then we have also tested some functional characterization using doxorubicin as our test model to test the IC50 value. We found out the using the combination gives us using polympholite with DMSO gives us very similar IC50 value, which is comparable with the control. And also we tested the cytochrome P40, P450 enzyme, CYP3A4, to see its functionality of the HepG2 cells, and because this is a liver model, so we wanted to test the how they are actually functional or not. So it seems there is a decrease in functional value, but it is not as bad as 10% of DMSO. So it can be used uh, for the study. And you need to remember one thing that uh, uh, these HepG2 cells are not good model for the cytochrome P450 because these are cancer cell line. So if you want to test properly, you need to do primary liver cells. Next. So the whole process, you might ask why you want to do cryopreservation of spheroid. The main intention of our of doing spheroid cryopreservation is to they be accept the ethical issue. That is important thing, but another is to decrease the time required for you to get your spheroid for your experimental purpose. If you do from the beginning, like take the cells and then thaw them and grow the culture them and make the spheroid, it might take you 7 to 14, which is realistically very less time. It might goes to 28 days to get a functional spheroid. So suppose you made a spheroid and then you wanted to do your experiment. You are waiting for some molecule that will come after 2 months, so you need to do it again. So using the cryopreservation of spheroid, you can reduce using our method or using any method, which is you can reduce the spheroid availability within 3 days. And we show that with our method, you can with polympholite and 10% DMSO, you can easily reduce the period to 3 days. And why we choose polyamphylite? It gives us compared to the traditional 10% DMSO alone, it gives us high recovery, high viability, intact membrane, and off-cell usefulness. So continue to this study. We also tried with RBC, red blood cell. There are a lot of paper on cryopreservation of red blood cell using trehalose and glycerol. But the inherent problem with glycerol is that it takes a lot of time to deglycerize the sample compared to DMSO, which is toxic, but it is easy to remove them using 2-3 time washing. So we try to cryopreserve the RBC for the sheep blood. We don't have ethical permission to do human blood. We tried with sheep blood and tried to cryopreserve them in combination with trehalose at with DMSO, not glycerol. We saw that when you combine polyamphylite with trehalose and DMSO, the result is recovery is around 90%. So the main advantage of this RBC, the RBC, these are non-nucleated RBC. So cryopreservation process is quite simple, and the permeability of different component is easy compared to the avian blood. If you can guess, these are two different type of RBCs which are shown here. The left hand side is the normal RBC, the non-nucleated RBC, which are generally found in mammalian like human or all the primates. But the right hand is the avian blood, which is quite different from in nature. They have a nucleus, and it has It is more like fibroblast cells. It has a nucleus, it has microtubules and intermediate filament. And the cryopreservation we using traditional glycerol method is quite ineffective in case of avian erythrocyte. There are different causes are there. They are more sensitive. They are more prone to osmotic stress and to remove the glycerol or any cryoprotectant, you need to do a lot of steps, and during that process, they lost their viability or integrity of the cell membrane. So remember this figure. I will come to later with this figure. So you can see there is an intermediate filament in all nucleated RBC. So we try to use a different combination of cryoprotectant to see which combination or which cryoprotectant is good for cryopreservation of avian RBC. For that, we choose chicken blood RBC, which generally available in commercially available in Elsevier solution. We took that and removed the solution and washed the RBC and combined with different combination of crop protectant. We put them in the minus Mr. Frosty and directly place them in -80 to crop preserve. The process is very simple. We generally use for mammalian cell culture. We tested different combination of crop protectant, only 60 mg of polyampholyte or only 4.4 molar of trehalose or only 10% DMSO. So above all, we found out that with all the permutation and combination, all the experimental setup we did, we found out that the combination of 10% DMSO with 60 mg of polyamphylite is good, give us a very consistent recovery. It is more than 80% recovery, and also we tested for after 14-day post thaw, but I didn't put. We also crab preserve them for 6 months. The results are similar. There is we are getting recovery more than 80 to 90% using the combination of 10% DMSO with polyamphylite. So, as I mentioned remember the figure of nucleated RBC, you remember I told you about intermediate filament. In control cell, these are we stain the cytoskeleton of the RBC. In control, you can see the intermediate filament, which are bright green color, which is very prominent in control. But when you cryopreserve the cells with 20% DMSO, 20% glycerol, we added also polyamphylite. We don't see that. The membrane seems to be very intact, but it loses the intermediate filament or cytoskeleton. Compared to combination with 10% DMSO with 60 mg of polyamphylite, it is quite good. You can easily recognize the intermediate filament. So again, we tested morphological integrity using SEM to see whether they are actually how they look like in real time. It seems with combination of polyamphylite, the membrane is very intact and it remain compared to other method we used. We tested post-apoptosis. As these are nucleated cells, the post-apoptosis is one of the key important factor which we look for. Here we have stained the RBCs with annexin V to look for the phosphatidylserine, which is out of the membrane. It seems that in case of as from other results and also this the 10% DMSO with 60 mg of polyamphylite has less apoptotic cells after 6 hours of post-thaw. So from all this study, we found out that there is something going on. We need to know more about that. So we try to answer this question using Microscope. We know microscope is one of the workhorse for the cryobiologist. It is easy to do the experiment with microscope, but you there is a limitation of data that you are getting. Uh, you might get where the ice formation started in a sample, and how they move in a tissue or a cell, how they propagate throughout the cell. You can look for that, but overall, how much intracellular ice formation, how they are say, distributed in the cell, you cannot get the information from normal microscope. So we try to integrate the cryomicroscopy with Raman. We are not the first to do this. There are groups who have already did this kind of experiment, but we try to get the information or the from the literature. We try to follow that, and we also wanted to see how polyampholyte works. So we tested different type of cells. It is very. I wanted to say that it is very difficult to do the Raman mapping with adherent cells. But when you take the cells which are non-adherent, like THP cells, which is one of the cells which used for T cell therapy, which can be used as a model for T cell therapy, we tested that cell, and from our study, we found out that when you use THP cell to see how the DMSO is goes inside a cell, you can easily identify the DMSO. Only DMSO, 10% of DMSO with 1 degree cooling rate, it easily penetrate the cells at around -50 degree. And but also you can see there are intracellular ice also forming during the cryopreservation. We know that intracellular ice formation is bad, but there are contradictory results are available, or literatures are available, which shows that cell can recover from the intracellular ice. I'm not saying that intracellular ice is bad for cell. Obviously, it's bad, but cell can recover from intracellular ice. Also, we tested with DMSO in presence of polyampholyte, the polyampholyte that we use in our experiment. It seems that the cells, as DMSO can easily penetrate inside the cell, and but when you look on the ice phase of the Raman spectroscopy of ice, there is comparatively very less, or maybe zero ice inside the cells. And we also look for the map of the Raman map of the polyamphylite. It seems they are, as we know, the polyamphylite cannot penetrate inside the cell, but they are forming a coating on the cells. It seems that they actually inhibit the ice growth. We also tested chicken RBC. They are very small, and also it's hard to get the exact size of them. But we try to use different image process to find out the before and after cell, and try to locate them, and then do the analysis that we did for other expert THP cell. We found out that the amount of intracellular ice is compare in case of 10% DMSO with 60 mg of polyamphylate, which is the best combination that we used, has less intracellular ice formation. How it works, from all the experiment that I showed, or some of the experiment I didn't show you, it seems that the using polyamphylite in combination with DMSO decreases the area of the cell size, or it sinks the cells comparatively higher compared to the DMSO on alone. And there is a chance the we also saw that there is less intracellular ice, which mean the cells are able to cope with that. If there is less intracellular ice, the cells can recover from them. And also from confocal imaging, some of the data I didn't show you, we found out that the fluorescent intensity, like which is a marker for intactness of the effect in cytoskeletal, is less damage, or there is less fluorescent intensity change, which might signifies that the membranes are quite stable in presence of the uh, polyampholyte. Uh, I think that's all. This is our group. We have more vacancy. If anyone wanted to join our group, they can apply or contact Matt. Thank you all.
Thank you, Dr. Bissoy. Great talk, great talk. And so we will take any questions from the audience. You can enter them in the Q&A chat, or maybe even raise your hand and I'll be able to unmute you.
Yeah, that will be great.
And then maybe I can just get us started. So it looked like when you were looking at those Raman images and you were looking at the ice formation and the glass formation, your formulation with the polyampholyte, it almost looked like the cells were smaller. Yeah, this. Do you
Yeah.
think it's like an osmotic effect, maybe, or do you think it's like solely attributed to the polyampholytes?
Obviously, there is osmotic effect, and also also polyampholyte. We check the change in the osmomolarity. It seems the polyampholyte doesn't has much more effect in changing the osmomolarity of the CPA solution. So it might be the effect of polyampholyte.
Thank you. Thank you. Uh, Francisca, I'm going to try to allow you to talk. Maybe you can ask your question.
Hello. Very nice talk. Thank you. Very interesting. Maybe you mentioned in the beginning, and I miss it. I don't know. But these polyampholytes, they are like easily accessed, like you can buy it, or you need to develop specific for your biological material that you are using.
This polympholytes you can prepare. It's a single step method, and also if you want, you can contact Matt. He is always ready to eager to help you out. And it's a single step process. You just need ether methyl anhydride and also dimethyl aminoethanol, and simple step. And if you prepare it, 1 kilo of polyamphylite will cost you around, we calculate, like Matt calculate, it's around 56 kilo, 56 pounds. So it is cheap. So,
Yeah. Wow. Nice. Okay.
and there are different papers are available from our group, so you can easily follow them.
Okay. Thank you.
Thanks.
Okay, we have a question in the Q&A box here. Hi, AKALABYA. This is Estefania Paredes. Nice presentation. Two quick questions. One, I was wondering if you have any idea how the polyampholyte will behave in a high ionic environment like seawater. And then also the question about osmolarity, but I think you kind of answered it with it being a polyampholyte doesn't change osmolarity too much, but feel
Yeah.
free to touch on that.
Uh, high you mentioned about
Oh,
high.
high ionic environment. So if you have a lot of salts, do you think the polyampholytes will behave the same way in regards to like
Yeah,
cryopreservation?
I think it will not precipitate. Probably you are talking about precipitation. I don't think it will precipitate, and it will probably it will work same as we think. Like now it's working. So uh, we need to test it. From my knowledge, what I know, it might not affect it. But if you have any certain uh, thing that you want to test, I can go for that. We can look for that.
Very cool. All right, Gurjit, I'm gonna try to unmute you and you can ask your question.
Thanks, Charles. Excellent presentation, Dr. Bissoy. I have a bunch of questions. Very good and excellent data here. One question is how fast the polyampholytes are able to penetrate inside the cell when you mix them with the CPAs.
And actually, I can answer in two way. Like this is not penetrating cryoprotectant by any means. So you can incubate for 10 minutes. That's enough for give you a uniform layer of cryoprotectant or say polyamphylite for doing cryopreservation. So one more data I can show you if you allow me to, so, which I thought might be interest for you. Yeah, this, if you can see it, like
Yes.
we use spheroid for cryopreservation of using polyamphylite. It seems that when you use spheroid and polyamphylite solution, they can penetrate in the spheroid, but for that we use FITC conjugated polyamphylite to see how they can goes inside the spheroid. So we you can easily find out they can penetrate inside the spheroid, but they will not penetrate inside the cells. So
Interesting.
did it answer your question?
Yes, actually, it's a follow-up question on steroids. Based on that, is like how like do you ensure that even when this whole solution is mixed, and I'm assume you said you mix and put it in Mr. Frosty for minus
Yeah.
80, and there is a certain degree how the temperature goes down, like 1 degree Celsius per minute. Like how
Yeah.
you ensure this reaches the I see the data and see the live and dead cells, but how do you like ensure does you mix them to make sure that this whole combination reaches the center of the spheroid, since reaching in the center of the spheroid is issue with the spheroids depending on how
Yeah.
big the spheroid is.
Yeah, this result will answer your question. You need only 15 minutes to, if you incubate your polyamphylite with spheroid for 10 to 15 minutes, you can polyamphylite can easily penetrate inside the core of your spheroid. And once you are done, you can cryopreserve them. And when wanted to remove it, with 10 minute or like 2 time washing, you can easily take out the polyamphylate. So it is easy to get inside the spheroid and also removing it from spheroid.
Excellent. Just one last question. I don't want to take all the time. Quick question on the Raman. Can you quantify the intra? You talked about intracellular ice. Can you
Yes.
quantify between the one with the polyamphylite and one with, let's say, no polyamphylite cells? Like, can you quantify the ice, intracellular
Yeah,
ice through that?
yeah, we quantify the ice. If you can have time, I put the paper probably in the citation. We already have quantified it using we from our group, one PhD student. He developed a Python program to analyze the post data and to quantify the intracellular ice. So it's there actually. You can
Great.
look for.
Yes, I'll follow. Thank you so much, Dr. Bissoy. Thank you, Charles.
Thank you, Dr. Bissoy. Great presentation. Thank you for answering those questions. You have a couple more questions in the Q&A, but due to time constraints, we are going to move on.
Yeah.
So I recommend if you have time, just going in there and typing out some of those answers.
Yeah,
But I'm going to hand it over to the president of ICYR, Dr. Laksha Gangwer from UMN, the University of Minnesota, to introduce our next speaker.
Thank you, Charles. So our next speaker would be Dr. Ivan KLBIK. He's a postdoctoral research fellow in Institute of Physics at Slovak Academy of Science. He obtained his bachelor in physics, master in biophysics, as well as chemical physics from Comenius University in Bratislava. And he did his PhD as well in the same institute where he's a postdoc. His research focuses on fundamental cryobiology, such as the mechanics of cryoinjury, cryoprotection, and currently he's focusing on membrane ion transport, and he will talk about that using the molecular dynamics simulations. So you will see a different presentation, a lot about computational modeling. The floor is yours.
Thank you very much for this introduction. So let's wait for the presentation to load up. Okay, so as you can see, I will try to tell you something about ion transport and its possible involvement in cryoinjury. We can go to the next slide, please. So basically, to sum it up, in case if there is someone in the audience that is not familiar with this, basically this slide is summarizing the Mazur's two-factor hypothesis of cell cryoinjury during freezing. Basically, on one hand, we have intracellular ice formation associated with high cooling rates. On the other hand, there is a slow freezing injury or solution effects injury when cells are frozen slowly, when ice is avoided, intracellular ice. Of course, there are other sources of cell cryoinjury, such as extracellular ice formation, also eutectic salt crystallization, and osmotic stress. Basically, we always need to add some sort of cryoprotectant, such as dimethyl sulfoxide or others. In this presentation, I will mostly deal with the osmotic stress, but also later a little bit with ice formation. So it was shown that basically osmotic stress is to a large extent composed of the injury that can be called post-hypertonic lysis. We can go to the next slide. And post-hypertonic lysis is a mode of cryoinjury that is typically observed for red blood cells, granulocytes, spermatozoa, adenocarcinoma cells, or Chinese hamster ovary cells. And in my experience, I have observed it also for skin cells, Hakat keratinocytes. So basically, the idea is that when cells are exposed to high ionic strengths, high salt concentrations, something happens which introduces this injury that can be observed most often only at the resuspension stage when cells are first they are exposed to hypertonic environment and then they are returned to isotonic media. On the graph on the right side, you can see that the cell viability is lost only after the resuspension to the isotonic media. Basically, mostly with the red blood cells, there has been a lot of research done on these cells, and there were cation leaks observed. So these cells were accumulating salts. In typical cell, this would mean that sodium chloride would be entering cells and accumulating intracellularly. What is typical for this behavior is that cells swell in isotonic media. Also, unexpected freeze-induced swelling was observed for some sort of epithelial cells. It is also known, especially from the research on the red blood cells, but also on these PC3 cells, that the extent of the injury depends on salt concentration and time of exposure to high concentration of salts. Next slide, please. So, in this presentation, I'm trying to cover the physiological perspective on these problems. And in one paper of mine, I try to provide explanation for this behavior. And I hypothesize that, in fact, the salt loading is caused by ion moving through ion transport pathways, which are naturally present in all cell membranes. Cell physiology recognizes cell volume regulation strategies of cells. Pretty much all cells possess these mechanisms, and basically the most abrupt cell volume regulation is done by activating ion transport pathways. There are regulatory volume increase responses as well as regulatory volume decrease responses. In typical cells, accumulation of sodium chloride leads to regulatory volume increase response, and on the other hand, there is loss of intracellular potassium chloride. Next slide, please. So in that paper, when I was hypothesizing that this mechanism could be involved in cell cryoinjury, I did some computational modeling. In this case, it was solving differential equations for ion flux through membranes, and there is a comparison between these two categories of cell responses. On the left side, you can see what happens if you increase sodium conductance through plasma membrane. Cell volume eventually reaches the hemolytic volume. On the other hand, there is the response of when potassium conductance is increased, the intracellular ions are flowing out of the cells. So these are just to show what kind of behavior can cell exhibit. The typical assumption in cell modeling is that salt conductances through plasma membrane are very low. This is often appropriate assumption when under normal conditions, but we have to consider that cryopreservation includes very high stresses like hypertonicity, which can induce these responses. Next slide, please.
Can you see the next slide, Ivan?
Yes. Okay, so just the summary of the proposed mechanism. So basically, during the hypertonic phase, these ion transport pathways are activated, allowing a flow of extracellular ions into the cells. The intracellular salt contents are increased, and then cells swell during the resuspension to isotonic media, and there is a possible lysis. We can go to next slide. So when I was thinking about this possible ion transport mechanisms involved, there is a summary of these. There is basically sodium, potassium, chlorine cotransporter, sodium proton exchanger, chlorine channels, also possibly the chlorine bicarbonate exchanger, and a bunch of hypertonicity induced cation channels. So this is based on some physiological studies. We can go to next slide. At this point, I was asking questions about what are these standard cryoprotectants doing? Like, we of course know that DMSO, glycerol, and similar agents reduce ice formation. They depress the ice freezing point, etc. Also, they decrease the salt concentration at a given osmolality. But I was asking myself, like, could it be the case that these agents could inhibit this ion fluxes through membranes, perhaps by some interactions with the membrane protein constituting the ion pathways? And answer to that question is, at least in part, yes. DMSO was found to reduce conductance of cations in red blood cells, which is very convenient for this hypothesis, let's say. And also on the right side, you can see results for chlorine fluxes in some other cells. And you can see that already quite low concentrations of DMSO reduce the chlorine conductance quite considerably. So we can move to the next slide. So I was trying to think about this, how to go about it, and I wanted to look at the mechanism. How does DMSO affect the ion conductance? So of course, there, at least in principle, is a possibility to look at the red blood cell channels, but these are quite complicated, and it is not known which exact channels are actually affected by DMSO, so it would be quite difficult to study those. So I went with the model potassium channel, which is found in bacteria. Uh, it's also a very nice channel to study in molecular dynamic simulations because there is plenty of published studies on that, so I could pick up there. So just very simple or very quick summary of this setup. I studied 0 or 10 vol% DMSO. The potassium channel was embedded in DMPC membrane, and there are some other details. It was also a potassium chloride solution only because this is for potassium flux. And yes, I used Gromax, and if you are interested, I can share more details if you have questions, so we can move on. So the most straightforward setup when dealing with computational electrophysiology is to use double membrane setup, as you can see on the right. It's very handy because it creates physically separated two solutions. So basically, one in the middle is, let's say, intracellular solution, and the other part is extracellular solution. And the membrane potential can be set up by ion imbalance within these two compartments. I done that, and I used quite high membrane potential of 700 millivolts. Of course, it's rather non-physiological. It was done due to statistics reason because in molecular dynamic simulation you can cover only short times, and you are kind of fighting with that you need some permeation events to make any claim. So yeah, we can move forward. So for the results, I observed about 20 picosiemens conductance in DMSO-free environment, and then when DMSO was introduced into the system, there was a significant or considerable reduction of the conductance. It was about 11 times lower than DMSO-free system. So yes, these results suggest that DMSO in fact reduces the conductance also in this channel, and we can move forward to look at the mechanisms. In this slide, I'm just showing partial density profiles that you can see these little peaks around -5 or +5 nanometers. So these are the sites where ions are sitting within the channel, and it's gonna be handy later in the presentation, so we can move forward. You can play the video. So I was trying to first look at what is the effect of DMSO on the structure of this potassium channel, because it could induce some sort of conformational changes that would lead to closed conformation, for example, but this is not the case. The effect of DMSO on the protein structure is quite small, and in this respect of ion conductance, there was no large effect. So we can move forward. On the other hand, there is a cavity within the ion channel, and DMSO, represented by yellow spheres, enters the cavity. The red spheres are the potassium ions, and it is quite interacting with the potassium ions within the channel and blocking it for other potassium channel coming in contact and knocking on those ions and making the transition. Next slide, please. So before I show you another slide, I just want to emphasize that DMSO is mostly present in the solvent phase, so it does not accumulate within the hydrophobic membrane, except hydrophobic hydrophilic interface, which is at the interface of hydrophobic core and polar head groups of lipid molecules constituting the membrane. Next slide, please. So on this slide, there are two DMSO peaks on the left graph, which are associated with the hydrophobic hydrophilic interface, but the peak at the very center, red curve, that is the cavity. So there is very close contact with of the DMSO and potassium ions, which matches the radial distribution function first peak, which is saying something about direct interactions of these atoms. So this is stabilizing the DMSO at the selectivity filter, the most narrow part of the ion channel. Next slide, please. So I'm just moving to other part or next part of the presentation where I promise you that I would also cover the ice formation or cryoinjury induced by ice formation. When I was doing some simulations of plain water sodium chloride solutions, I observed that ions get entrapped to some extent within ice crystal lattice. And I was thinking about it because I observed that chlorine ions do get entrapped at higher rates than sodium ions. And I was thinking about it from the perspective of membrane potential, that this produces charge imbalance, which is actually dictating the membrane potential, and it could and it would affect it. So then I found that there is Workman-Reynolds freezing potential, as described previously. Is an effect that describes this behavior, that there is actually quite significant membrane, or sorry, electric potential buildup during freezing of electrolytes, and this could be actually involved in this cryoinjury. So we can go to next slide. Here I just show some comparison that the force field that I used could to some extent describe the reality, because it's also observed experimentally. We can go to next slide. So basically I tried to observe what is the effect of DMSO on this ion entrapment rate, and I observed that DMSO actually reduces the ion entrapment rate, which would be considered as detrimental from the perspective of the cryoinjury. So this could explain some aspect of DMSO cryoprotection. Regarding the mechanism of DMSO action, of course, there is less ice when DMSO is present, so less opportunity for ion entrapment. And also DMSO considerably slows down the ice growth rate, so there is more time for the ions to diffund away from the interface. Next slide, please. You can play the video, Charles, please. And I tried to do simulations with this double membrane setup, and you can see the ion entrapment within ice crystal lattice. Because there is a possibility for electroporation. If the produced membrane potential is very high, it could actually induce membrane poration, which could be fatal for the membrane. It did not happen. I observed membrane potential of some 300 millivolts, which is quite a lot. And in the case of extracellular ice formation, it translates to hyperpolarization of membrane, which could in fact induce apoptosis. So it is not enough for membrane poration, but it's enough for affecting the cell function or inducing apoptosis. So we can stop here, and just to sum it up, I can say that ion transport should not be overlooked within the cryobiology, and the importance of cell physiology perspective should be should be appreciated enough. Thank you very much for your attention.
Yeah, very nice talk. Thank you, Dr. KLBIK. If anyone has question, please raise hands. I see there is one in Q&A box. Maybe we can start there. So it's from Dominica. He says, hi, Ivan, your finding seems to be especially important in neurons. Do you think we could externally regulate the ion concentrations in anticipation of the channel changes? Or should we look at small molecules temporarily channeling the channel properties?
Mm, And let me read that. Mhm. Yes. Well, there are possibilities like using ion channel inhibitors. There is quite plenty of them, and many substances are well characterized in this respect. Um, for example, for these channels, amyloid or gadolinium ions or flufenamate and substances like that. Um, or try other agents. But it is very important to emphasize that the effect of the DMSO is gonna be on a different level than these inhibitors, which are introduced at very low concentrations. DMSO would be needed at higher concentrations. Like okay, 1 or 2 or 3 % is not a lot, but these specific inhibitors require like 100 micromolar or something like that.
Thank you. I see Abhilash hand is raised. Yep, Abhilash, you can unmute.
Hi, Evan. Thank you for such a nice presentation. I had a question on the DMSO ice growth part. So, the ice growth, when we add DMSO in real life, it's like very slow. So, first thing is that what concentration of like water and DMSO did you work at?
I studied DMSO concentrations up to 10 volumetric percent.
Okay, so then you must have like ran the simulation for like long time, right? And how much was the time that you ran it for? And
Oh
because
yes, typically something on the scale of 1 microsecond or 1.5 microsecond, something like that.
Oh, that's a huge time. Okay. So when this thing happens, so then some of the DMSO molecules are trapped in the, like you showed in the ice crystal. So it will change the concentration, in the water as well, and that will further slow the growth. So did you see that?
Um, these pictures I was showing, it was ions getting entrapped within ice crystal lattice. I did observe some entrapment of DMSO, but it was like 1 or 2 DMSO molecules, so mm, it was negligible. It was negligible. For the ions, it was from the osmotic perspective, I could say that it is kind of negligible. You
Mm-hmm.
can kind of forget about it, but from the perspective of membrane potential, it can be very huge because it was like, for example, experimentally 100 millimole solution of sodium chloride, something like 10 millimole get entrapped within ice for yeah.
Okay, okay. And one more question, like on the your barostat that you said and the thermostating. So you said that you're using the velocity rescaling one there. So
Yes.
I mean, I know velocity rescaling, it will just adjust the velocity at every time step, and velocity decides the temperature and other properties. So, but in general, people generally use NVT or Langevin thermostat or other thermostat. So what was your reason that you used rescaling one? Because it's a pretty harsh thermostat. And maybe did you try to benchmark the simulations with that with the some properties and with DMSO water so that you know that the thermostat is correct and the force field is also correct?
hmm Actually, we tried a couple of different approaches. When it comes to barostat, uh, but when it comes to thermostat, actually the VIRI scale is recommended for the amber force fields. I'm aware that with charm force fields, it's different story, but for amber, it's the most standard option actually. Yes, there is a stochastic term for the regulation of yeah, introducing some sort of chaos term, but for Ember it's quite not quite. It's actually the standard thermostat.
Okay, yeah, that's all. And thank you. And it's really nice work that you did this on this DMSO and water thing.
Thank you.
Hey, I think you have one more question in the chat Q&A, but you can answer that privately. And we'll move on to our next speaker. Charles, you want to introduce?
Yes, sir. Yes, sir. I am excited to introduce our next speaker, a good friend, Dr. Troy LOUWAGIE. Troy is an R&D scientist at Evia Bio. He graduated with his PhD in December of 2025 from the University of Minnesota in the Mechanical Engineering Department. At the University of Minnesota, his work included optimizing DMSO-free solutions to improve cryopreservation processes of cellular and subcellular systems. At Evia Bio, he works on developing and optimizing solutions for mammalian cell types towards commercialization of cellular therapeutic products. Today, Troy will be talking to us about cryopreserving Drosophila melanogaster embryonic nuclei in a DMSO-free solution utilizing some computational optimization methods. Troy, the floor is yours.
Great. Thanks, Charles. Let me share my screen here if I can figure out how to do that. Okay. Can you see my screen?
Yes, sir.
Great. Okay. Yeah, my talk is on the cryopreservation of Drosophila melanogaster embryonic nuclei in a DMSO-free or Me2SO-free solution. I forget that, this was for Society of Cryobiology, and they prefer the Me2SO-free. I'm definitely going to be changing it up in the presentation. You'll see DMSO-free as well, so I apologize for that. But so just background on Drosophila melanogaster, also known as, the common fruit flies. So these are model organisms used for biological research. They've been used to study diseases, genetics, and other various research topics like circadian rhythms, for example. And just generally, these fruit flies are maintained by continuously flipping cages of live fruit flies so that they produce the next round of fruit flies. And so there's no form of large preservation of these systems. And so you have these large stock centers in Bloomington, Kyoto, and Vienna that are just, their roles are just to keep specific strains of fruit flies. And so with these, you have just these costs of maintaining populations, let alone, the labor that is needed to keep these specific strains available. But you also have genetic drift and contamination. So just one story is that we ordered a specific strain from one of these stock centers, and it's an easy strain where embryos have a green fluorescent tag to them, and when we looked at them, they didn't have that tag, and so they some of them were green, some of them weren't. So you've got issues of contamination just let alone in those stock centers. So we're really trying to find a way for preserving these stocks. So there has been some work on cryopreservation of Drosophila strains. In the 90s, there were methods looking to vitrify these embryos. In 2021, a group from actually the University of Minnesota as well, and John Bischoff's group improved this vitrification method of embryos. And then there's also a group in Japan that looked at vitrifying primordial germ cells for transplantation. And so, the method that we're going to talk about today is a slow rate cooling method for isolated nuclei towards a transplantation method. So just briefly, I want to go over, we all know this, or we should all know this as cryobiologists, but, metrics of cryopreservation, right? We have recovery, viability, and function are kind of our three main methods of cryopreservation. So recovery is the number of live cells divided by post-thaw divided by the number of live cells that you have pre-freeze, in this case nuclei. Viability, number of live cells divided by the number of total cells, and function, right? So in cells we have, with CAR T cells or natural killer cells, we have these cytotoxicity assays where you can put them in co-culture, and you can look at how well they or how effective they kill cells. An easier functional assay is just proliferation. So, is it performing what it should be performing after freezing and thawing? So why do we care about fruit flies, and specifically why do we want to do nuclei from these fruit fly embryos? So like I said before, they're a model organism, and they're just widely used across genomic research. And so for nuclei, these eggs are laid, and for essentially 2 hours, these nuclei are dividing every 7 to 8 minutes. So, in a short time span, you can get thousands of nuclei just from one embryo. So if you're able to collect and pool nuclei and cryopreserve them, you potentially have a large stock of a strain. And then a potential way to preserve this stock is to cryopreserve the nuclei and then put them into a host embryo to then develop a strain. And so this figure in the bottom right is just, it's a slow capture of just the uniformity of nuclear divisions. And, it doesn't mean anything to cryopreservation, but I just love the beauty of, these divisions that happen so uniformly. And so then, why do we care about DMSO-free cryoprotectants? So DMSO is a typical CPA used, and nothing against DMSO. It's been the gold standard and still is the gold standard, but it is toxic if it's left in the solution. For thoughts of injection with these nuclei, we wouldn't be able to remove the CPA. So we wanted to look at DMSO-free cryoprotectants to avoid the toxicity. And so in our lab at the University of Minnesota, in the Biopreservation and Core Resource Lab, we've been able to find combinations of sugar alcohols, and amino acids that can replace DMSO entirely. And this has been used for multiple cell types. We've been able to cryopreserve Jurget cells, iPSCs, neuronal stem cells, and mesenchymal stem cells, all by optimizing these 3 components. And the way that we optimize this is with a differential evolution algorithm. This is more of an optimization algorithm than machine learning. But so you start out with essentially a span of solutions, different components that you want to test. So in our case, we want to look at 11 different levels of 3 different components. And so that's 11 cubed, different combinations if we wanted to test all of them, which is just too many. So what we do is we set out with, an initialization of a random vector space. So, we can start out with, these levels 5, 3, 3, 1, 4, 9, and 10, 2, 2. And then what happens is you test those components, and you can, test based on initial recovery or viability or 24-hour proliferation. With the nuclei, we optimize based on recovery. And so then essentially what you do is you mutate that vector. So if we're looking at 533, right, by mutation and crossover events, you can now change it to, instead of 533, it becomes level 556 to test. And you do that for the other initial parameters too. And then you test that mutant set of parameters. And then you select the top half of those. And then you repeat that process, and you repeat that over and over until you've gotten your output, which is, optimize recovery. So this reduces the total number of required experiments to find optimal concentration. With the nuclei, we did this differential evolution algorithm, and we did a 5-level DE algorithm with sucrose, glycerol, and isoleucine. And in 4 generations, we were able to find 5 DMSO-free solutions with greater than 80% recovery. So again, this is 6 cubed. So quick math, I think that's 218 possible solutions, and we did it, in 4 generations, which is 5 experiments. So, in a quarter to a third of the search space, we're able to optimize these concentrations very quickly. And we also did cooling rate and nucleation optimization, where we found improved recovery using a cooling rate of 3C per minute. And so then, you probably didn't think you were going to hear 2 Raman talks, but you're getting 2 Raman talks today. We used Raman spectroscopy to look for intracellular, or in this case, intranuclear ice formation. And so just a brief background on Raman spectroscopy is that, a light source interacts with a sample, and most commonly you get Rayleigh scattering. So, the energy in is the energy out. With Raman scattering, what you get is when the light source interacts with the sample, you can have, essentially the bonds are being excited to perform something. So in water, you can have OH bending or twisting of these bonds, and they release a much smaller energy. And so this is the Raman scattering. And so by using a grading system, you're able to essentially remove the Rayleigh scattering from being measured and identify bonds of specific molecules. And so, in this case here, this is, 3 different peaks of DMSO, cell, and ice. But we were also able to, find specific peaks for the nucleus and non-DMSO components too. And this is also the setup. It's a little bit differently than the one that the Gibson group uses. But what we have is a Raman microscope and a Peltier module on top of a cooling stage. And essentially you attach a temperature controller to the top of the Peltier module where you put your samples on. And then you can control the cooling rate with that temperature controller. We also surround the environment with we create an airtight environment, then purge with nitrogen gas so that you can, you don't have to worry about ice formation, ice condensing on top of the sample. And so with that setup, right, we were able to image nuclei and observe that, there was no intracellular ice or intranuclear ice inside of the nuclei during freezing. Which was also really good to see. But now coming back to, what is success for Drosophila nuclei in cryopreservation? So we come back to these 3 metrics that I talked about earlier, and that's, recovery, viability, and function. So recovery, we're able to develop a method, just the recovery was the number of intact nuclei that were counted post-thaw and compared to the number of intact nuclei that were counted pre-freeze. Viability is not really an assay that can be performed on these systems because you know, a bunch of these viability stains are nuclear stains like AOPI or DAPI. So all of these bind to the nucleus. So you can't do a viability stain with nuclei if everything binds to the nucleus. So then we come back to, we need another metric to make sure that these nuclei are being frozen properly. And that's where we come to a functional assay, and we ended up using reverse transcriptase, quantitative polymerase chain reaction, otherwise known as RT-qPCR. And this monitors the amplification of a targeted RNA sample. It's used in diagnostic tests. I know we don't want to remember COVID, but RT-qPCR was essentially the method that was used for doing all of these quick COVID tests. And in this case, we look at the heat shock proteins in nuclei. So essentially, you know, heat shock proteins are found in mammalian cells, they're found in Drosophila, they're found in a bunch of species. But if you elevate the temperature in a cell or a nucleus, the RNA starts transcribing. You know, these transcription factors are activated and these heat shock proteins start being transcribed. Or the heat shock gene, sorry, starts being transcribed. And so we're able to use, you know, relative quantification and compare the expression of this HSP70 gene compared to actin, which is just a universal substance. However, when we are doing these heat shock assays, what we saw in the top left is that, you know, just holding these samples for an hour, we saw loss of the heat shock function. And so that was concerning because, you know, if we don't have a robust pre-freeze method, we can't, you know, robustly use a post-thaw. And actually what we found out was we weren't losing that function. We were actually, you know, the heat shock was actually also being cold shocked. So we were holding these nuclei on ice to transfer to, you know, start freezing. And turns out that when these samples are held on ice, they also start transcribing these heat shock genes. And so the cold shock was actually inhibiting the heat shock assay. So we pivoted and measured activation based on cold shock instead. You know, one of the questions is if you're freezing these nuclei and you're doing a cold shock, how can you ensure that, you know, what you're measuring or what your function is happening, you know, is due to post-thaw function? Like, are these nuclei actually functional post-thaw, or are they just being cold shocked super quickly, you know, before freezing or during freezing? And it actually turns out the cold shock wasn't activated until you did a room temp chase or a hold. So as long as we didn't, you know, take them off ice or, you know, we kept them at low temperatures, that cold shock wasn't induced. And you can see that down below with low expression, you know, without the room temp chase. And so then we looked at this, you know, and so we did, you know, a pre-freeze heat shock where, you know, once they were held on ice and chased at room temperature for 30 minutes, they were expressing, you know, this cold shock. And then we also looked at freezing and thawing and looked at the expression of these nuclei post-thaw. And what we saw was clear function of these nuclei post-thaw, which was really exciting. And so, you know, we detected this both immediately post-thaw and 1 hour post-thaw too. So, you know, this was just really exciting to see, and clear evidence that nuclei were being frozen, thawed, and functional post-thaw. And so then this kind of leads to the you know, great, so we can freeze and thaw nuclei. What does that mean? And so this was a three-part project, which was really enjoyable to work on. And that's, I think, part of the fun of being a cryobiologist is you're working with multiple groups that have more knowledge of a system than you, but you're able to apply what you learned into that subject. So there's a group that created an auto injector, and this auto injector actually has been used to help vitrify zebrafish embryos. So this auto injector was created so that it could quickly inject nuclei into embryos. And then there's the cryopreservation aspect, and then there's the transplantation. So, getting nuclei into host embryos and then making sure that the embryos grew. And so that's kind of where we're at right now, is in the transplantation. So we're able to inject fresh nuclei into host embryos, and those nuclei are able to grow. So these are wild type embryos that have been injected with a GFP strain that can, the GFP strain is targeted in the nervous system. So you're able to see these like partial developments in these embryos. And so the clones do hatch, but the embryos, or the clones hatch from embryos, but then they die during the larval stage. And so the goal is to optimize, being able to inject these nuclei into host embryos, and then also do this with cryopreserved nuclei. So with that, I just want to thank, the people in my lab at BioCore, the innovation lab that does all of the fly work, and then also the funding agencies for this project, which were the NIH and then the NSF, which is used at the characterization facility. So I think with that, we can take some questions.
Yeah, thank you, Dr. Loug. We will take any questions, so feel free to raise your hand or put any in the Q&A box. I can get us started though. Was there any other functional characterization that you were able to do after cryopreservation of the nuclei? I don't know, I'm thinking a lot about like the nuclear pore complex, which is like this crazy thing that it like regulates what comes in and out of the nucleus. That was kind of one of my questions. Did you look at that at all or anything else?
We looked at, I'm trying to remember. So, I mean, the easiest functional assay would be to inject these nuclei post-thaw into an embryo and look at development. So that's, if that was possible, that would be the easiest one to do. We didn't do a whole lot of nuclear pore complex. I'm trying to remember. We looked at one other assay that I can't remember what we did, but may have been like intact, but I can't remember. And we really stuck with the heat shock assay just because we saw large expression in embryos, and we did see large expression in nuclei.
Thank you. We have one question. Thank you for the presentation. How are you doing the cryopreservation steps? So I think maybe this might be asking about the injection, or I don't know if you guys are incubating or however your cryo.
Yeah. So I definitely glanced over that. So we would isolate the nuclei. So essentially we'd collect, embryos for an hour or 2 hours or a couple different 1 hour flips. And then isolate the nuclei by essentially smashing up the embryo and spinning down and taking out all the debris. And then we would put in, so there's a specific buffer to isolate the nuclei, and then we would put in our cryoprotectants, and we do a 1-to-1 ratio. So we'd make our cryoprotectants at 2x the concentration, and then dropwise at it. And then we used a controlled rate freezer from Planer, which I think is now Next Spring Health. And so we did a controlled rate freezer, you know, and we looked at 1C, 3C, 7.5C per minute, and then we stored them in liquid nitrogen dewars.
Thank you. Then we have a question from Gurjit. Can you do metabolic assays on thawed nuclei for functional assessment?
I guess I don't know what metabolic assays you would do. We didn't look into that. I think, you know, the reason that we did this RT-qPCR is that the nucleus, the main goal of the nucleus is to transcribe RNA. And so if you're doing this, if you're doing RT-qPCR, you're measuring RNA transcription. And so that's, you know, if we're looking at RT-qPCR, we're looking at the main function of the nucleus.
Yeah, I think that makes sense. And but Gurjit, that's a great question. I almost wonder what the metabolome of a nuclei is.
Right.
That's really cool. All right, and then I guess just a final question: where do you see this technology going? Right, so almost maybe instead of like cryopreserving, or maybe like tough strains that can't be cryopreserved, you just preserve the nuclei, or what do you think there?
You know, so you know the cryopreservation, right? We call it a toolbox, right? So I think, you know, the current methods are pretty good. So, you know, Laksha's lab, also at the University of Minnesota, developed this cryo mesh method to freeze these embryos, and they do a pretty good job. You know, cryopreserving nuclei is just another you know, toolbox, another tool in the box to use, right? So I don't necessarily know if it's going to be used with Drosophila. There's been papers that they do nuclear transplantation for honeybees, right? So potentially could be used in that area. But you know, I think
Gotcha.
if there's the I think, you know, if you look at systems, you get, you know, a cell, and then you start going to spheroids, and you start going to tissues and organs. And so the way that you have to freeze can get more unique, or, you know, less general. And so a nucleus is potentially the most general thing in a cell. So, you know, it could be used for potentially other nuclei applications.
Thank you. Thank you. All right, let's give a round of applause to our speakers. We can't thank them enough. Some 3 great presentations by Dr. Bisoy, KLBIK, and LOUWAGIE. Thank you all for joining this ICYR-hosted Society for Cryobiology webinar. For those of you who don't really know much about ICYR or don't follow us on LinkedIn, I'm going to try to drop a link in the chat right now so you can go follow us and kind of keep up with what we're doing. We're currently having a leadership nomination. So if you go on there, there's a link to our ICYR leadership nomination form. We're currently giving away a $500 professional development award. That closes, I believe, on the 1st. So if you're looking to advance your skills in cryobiology, we have that money available for our members. We host a monthly ICYR Meet the Authors Journal Club. So if you follow us on LinkedIn, or if you're an ICYR member, we send out emails about our monthly journal club. Next month, it'll be about isochoric vitrification. And then just a recap on the annual meeting of the Society of Cryobiology, Cryo 2026. It's going to take place in Seattle, Washington from July 15th to the 17th. We have abstracts due on March 15th. Let me double check here. Abstracts are due on March 2nd, so get in your abstracts to present at Cryo 2026. We have student travel grants and awards due on February 19th. And then if you know someone you'd like to nominate for the Day Young Gal Young Investigator Award, that'll be due on March 15th. Laksha, anything I missed?
I think you covered it all. Just kind of wanted to say, please, if you want to engage more, so sign up for the ICOR leadership positions. And this was a great talk. All the three speakers, we appreciate them. And next year we'll have this webinar again. So wait for a year to see the ICOR hosted Young Cryobiology Researcher webinar. And thank you for joining.
See you
Right.
next time.
See
We'll
you.
host one again next year.
Yeah. All right. Thanks. Bye-bye.
Thank you
Thank
for
you again, Dr. Bissoy. Great talk.
thank you for giving the opportunity. It's nice to meet you.
Nice to meet you too.
We're looking forward for your research. Are you
Thank
giving talks in
Thank you. I actually have to give a seminar right now, so I'm gonna jump over for the Uniform Service University. But I'll be at Cryo 2026 if you attend in person or virtually, and we could touch base
Probably
there.
from our group, there are people will be there. So
Perfect.
not me this time. Maybe next year we can see together.
Yeah, no, that sounds great. That sounds good. Thank you again. Great research. Really excited to see what comes next.
Thank you. Bye.
Have a good day. Bye.